Hello everyone, welcome to Bioscient once again. I hope you all are doing well. So in my previous video, I was discussing about the biotechnology. There are already two videos of this topic. Now I am continuing that topic further. So if you have not seen those videos, you can see. I will put the link in the description box. Now, today I will be discussing the step which was there in the biotechnology. Those who are watching for the first time, I will tell you that biotechnology is a branch of biology where we are using technology on some living organism to produce the products which are important for the human welfare. So, we required some enzymes for this process, some vectors for this process. Today is the turn for the competent host. The host which enables the desired DNA or the genes which you are looking for to multiply inside them. So, how can we prepare the competent host? There are different ways by which we can make the competent host. First of all, we should know what are the preferable competent host. Like if we talk about the prokaryotic cell, the most favorite of all of us is E. coli. Do you know why? Of course, its multiplication time is just 20 minutes. So we will have multiple copies in a shorter span of time. So we generally use E. coli for our experiments when we prefer prokaryotic cell. Now we can have options of plant cell, animal cell as well as fungal cell. Again, in fungus we have priority of yeast, easy to handle with the unicellular fungus. Okay. Now, how many of you know that except animal cell, the bacterial cell, the plant cell and the fungal cell has cell wall? Now, if you want to insert your desired DNA or the gene in these kind of cells, you definitely need to break the cell wall first. Of course. So you have to remove the cell wall. We have discussed about this. How can you do that? Now, for now you should be knowing that we need to create a protoplast. You must be wondering what are protoplast. Protoplast are the structures when you remove the cell wall. Remove the cell wall, now you have what? Just the plasma membrane left with you. Okay, the protoplasm surrounded with the plasma membrane, now you have the protoplast. So, this is the cell without cell wall. The cell without cell wall is the protoplast. Now, when you have the protoplast, you just need to treat the plasma membrane with the calcium ion. Okay, the plasma membrane will be treated with the calcium ion so that it increases the permeability of the cell membrane. I hope you are getting brain. So why are we giving the calcium ion here? It will help the plasma membrane to more be permeable, more permeability will be there, which is actually desired for the purpose. Okay. Now, once the DNA is inside the cell or the gene is inside the cell. Now, what is our main aim? Of course, the cell having its own DNA need to be integrated with the your desired DNA so that when the actual DNA of the cell will be replicating, your desired DNA will also be replicating. So for that we give shock treatment, shock in a positive sense. So what do? The cells and the desired DNA is inside. Now they need to integrate. So we are just giving them shock, like you get shock, something weird happened, right? So what happened when the ice cooled, the temperature should be very low and suddenly you are heating up to 42 degrees Celsius and again you are giving the low temperature. Because of the temperature variation, the gene of interest and the host DNA will integrate. The problem is solved. This is what we want actually. So now we are ready with the competent host. So we have learned about the steps where we have created the protoplast, then we have treated the plasma membrane with the calcium ion, and then we give them shock treatment. Now, to proceed further, there are methods where you do not require any vector for the gene transfer. There are certain methods, and I must tell you, in the NEET exam, there are many questions you can expect from here, and it's already in PYQ, previous year questions. Okay, so now I'm going to discuss about the vectorless gene transfer methods. One of them is chemical method. I need not to tell you what is protoplast once again. That is required here and along with your desired gene or DNA. So both are kept in a chemical. That's why we are calling it chemical method. Both are kept in a chemical called polyethylene glycol. If you keep these two together, you need not to do anything. They will simply integrate. Okay, so this is how the chemical method works. And then we have a gun, not the actual gun. It's gene gun. We are dealing with very micro molecules. Okay, so gene gun or shotgun, the question was asked on this term biolistics. So this is also called as biolistic. Of course, a gun will do nothing without a bullet, isn't it? Let's make bullet. It is a bit costly process because you need gold. You know the rates of gold nowadays. So the gold, and if you are not able to purchase gold, you can take tungsten as well. So these small particle of gold, small in the sense it will be ready, even after getting ready it will be just 2 micrometer. So the gold particle will be coated with the DNA. Okay, now the complete set is called bullet. Okay, and now you need to shoot. So the speed, it's a weird, what is this unit? We have never heard this, so you have to memorize this. 1400 feet per second. Now, what if the speed is more or less? If it is more, like we say, we have 2 years, you have to hear from one side and you have to just leave that from the second year. It's not like that. So if the speed is more, the bullet will cross the cell. If the speed is less, what will happen? It will not reach to the nucleus. So the speed should be optimum for the process. Now coming to the third part, that's electroporation. Here we are using the electricity or the high voltage, very high voltage, but not for the very long time. It's for very short time. What will happen if you're keeping the cell in a medium and providing the voltage? It will create temporary pores in it, and that is the need of the hour. The pores is formed temporarily, so the desired DNA will get inside and the process is done. Okay, now last but not the least is the micro injection. I must tell you, it's a very important way, but it needs expertise. Do not try at home. So basically here you need a device, because if you are dealing it a very small size cell under the microscope, it would be really difficult. So you just need to hold the cell using a vacuum pump. This is the tube and this is the cell. Preferably it's a ovum, a human ovum. So you need to handle it carefully. So this vacuum will create a pressure so that the cell will be aligned. Now the job is not over yet. There will be a guide guide, which will be, this is the injection. This is nothing but the injection. This is the injection by which you are putting your desired DNA. So this red line is the guide and this purple line is the injection, which is directly focused into the nucleus, and that is how the micro injection works. This was about the making competent host in the biotechnology process and how do we transfer the gene without using vectors. I hope you have understood this fact, and for any query you can write down in the chat box. That's all for today. We will be coming with the new video on biotechnology. Till then, stay tuned. Thank you.